All right, let's talk about blood vessel, peripheral. We talk about cardiovascular system in relation to the heart and associated blood vessels. Today lecture is about peripheral blood vessels and circulation. So as we know, we have arteries that carry oxygenated blood from the heart throughout the body, and the smallest subdivision of arteries is arterioles. Those are the smallest ones that then come in join with venous end of the circulation, forming capillaries, where the actual gas, nutrients and waste exchange occurs. And here, as you can see, we have pinkish color, meaning that, again, I stress, the blood here is mixed as far as content of carbon dioxide and oxygen. On the other hand, capillaries on venous end continues on joining together, forming venulas, the smallest subdivision of venous end of the circulation, and then forming veins of different diameter and different size, finally joining into to a superior or inferior vena cava, or from the coronary circulation is coronary sinus. Blood vessels, the ones that we talk about, associated with the heart or heart attached, which include pulmonary trunk, aorta, and smallest blood vessels, and then, of course, pulmonary arteries and coronary sinus. This is just for your review. As I told you before, the blood vessels, just like heart, composed of three major layers. One is outmost layer, connective tissue, called tunica externa or adventitia. Adventitia and tunica externa is the same thing. It's connective tissue, mostly areolar connective tissues, that with intensive collagen fibers that link along the length of the blood vessel, protecting them from damage. Next layer, or tunica media, the central layer, or medial rather, layer is tunica media. Tunica means cover. Tunica media composed of the smooth muscles located circulatory around the opening, which we call lumen. And innermost layer of the blood, any blood vessels, is endothelial layer called tunica interna, and it's also composed of the squamous, simple squamous epithelial cells. The capillary size is the capillary size is diameter of one red blood cells. To give you an idea how small subdivision of the circulation blood vessels is. Veins, on the other hand, artery veins both have, this is a capillary, and it's shown how oxygenated erythrocytes come to the arterial part, and we'll get back to veins again. Then gas exchange happened here, and then carbon dioxide join erythrocyte on the venous portion of the equation. This is again tunica adventitia media and interna. Tunica adventitia or tunica externa is the same again thing. So you can, lumen is the opening of any blood vessels, just for you. The differences between arteries and veins that you will need to know and need to be able to identify looking at the slide, and it's not always a good clear picture as to how you differentiate between the two, but this is how they are present. And again, they all have the same layers, the same tissue involved in structure. So how would we differentiate the two? First of all, the arteries maintain their cylindrical shape. Again, function defines structure. So if artery pushes the blood all the way through the body, they have this cylindrical shape, pronounced, very serious muscle, tunica media, developed very nicely and pronounced forming large cylindrical shape. On the section, cross section as it's shown here, it could be a tiny little area like this if the blood vessel is at the moment contracted and pushing the blood through the circulation. Sometimes a nice and round opening. Vein, on the other hand, much more passive. They often collapsed. They have tiny layer of their muscles around, much smaller, thinner than artery have, although again it's not always the definitive criteria. You have to look for something else, which I will tell you for what. So vein here, the irregular shape of the lumen telling to you that it's actually vein, because they don't maintain the cylindrical, they don't push anything. So most likely they will have this irregular shape. What else? And usually veins and arteries located on the slide next to one another, and so do nerves or lymphatics. What else is interesting, and I'll show you the comparison slides in a little while, but if you look here, you will see like ruffled edges of the endothelium on arterial part of the slide, and nice and smooth endothelial here. So if you ever have any doubts about whether or not the blood vessels that you're looking at is artery or vein, you look at the edges of the opening of the endothelial layer, tunica intima. Ruffled, folding layer belong to artery. Why is that? Because artery have to contract and expand and regain the previous shape or basic shape. So they expand, the elastic. Therefore, in tunica adventitia, a lot of elastic fibers. Tunica media, which is muscles, capable of contracting and relaxing on its own, right? What is not capable of contracting and relaxing and expanding the size is endothelial tissue. Epithelial tissue does not have this capacity to expand in size and rebounds back. So, to enable the whole blood vessel on arterial portion of the circulation, artery, to be able to expand in size, stretch, and bounce back without any damage of any tissue, the folding or ruffling of the endothelial tissue is the key. So it allows the arterial innermost layer to stretch out following the expansion of the whole vessel and then bound back. Veins don't have this because they don't stretch out, they serve as a simple passage tubes which walk out of, like we discussed, skeletal muscle contraction. So this is how the veins and arteries join in through the capillary network. The other thing that veins, and sometimes it's possible to see when you're looking at the vein in microscope, because small microscopic veins also have this, it's valves. Valves, pockets of endothelial tissue that makes tunica interna of the vein. And just like in the semilunar or valves of the heart, it's allow blood to go through and then close in because blood tend to go down under the gravity. I will show you a few pictures I have there for illustration. This is how all tunicas look microscopically, just in a large magnification of the section of the blood vessel. This is longitudinal section, so you see nice tunica media here, vasculature, tunica adventitia, which is a regular connective tissue, and here you have tunica intima with endothelial cells. Well, difficult to say. For example, looking what else is important, elastic fibers inserted, embedded between the muscle cells of tunica media, typical for arterial blood vessel. They help maintain cylindrical shape, maintaining and turn the condition for blood pressure regulation, and they also allowing the stretching and rebouncing. So if we have elastic lamina here in smooth muscle, in this pronounced quality, it's most likely the arterial section. Also, again, you have to look at the cross section as well. This is what for typical for artery. Ruffling or bounce of the folding of the endothelial cells, and there is also subendothelial layer of connective tissue, which again in artery is mostly represented by elastic fibers, but also collagen. This is the typical artery with ruffled endothelial, roundish lumen, and a layer of smooth muscle. External, mostly adventitical collagen fibers. The other thing that blood vessels are also organs. Those organs require the innervation, which is nerve fibers next, lymphatic vessels. Lymphatic help to maintain interstitial space pressure, content. Vasa vasorum meaning the blood vessels, vasa vasorum supplying blood to the blood vessels. So the large blood vessels have the vasa vasorum that supply blood to them, just like anything, any other organ. This is comparison again of the artery and vein, and you see here ruffled or folded tunica interna. This is vein, and you see much smaller, thinner layer of smooth muscle, which much smoother, no folding, no nothing, tunica interna. But again, you're not gonna see picture like here all the time, and when we come to the lab, you will see that this is not always the case, so you have to look in all aspects of the structure, including the whether or not you have folds and how the opening is located. Vasa vasorum, we talk about this as a picture with very good impression. In the lab we have very nice slides that have side by side blood vessel, or the artery, veins, and nerves. So for you it would be a very good example. And don't forget about lymphatics that we're going to discuss much a little bit later, probably next week. This is comparison table I'm giving you just to memorize, like summary of the differences between arteries and veins. The same here we have, and the same another one. And here is very nicely shown the valves inside the veins with enlarged portion of the vein when the this part of the vein full with blood. This is again comparison slide, and this is comparison, so I'm giving you many comparison slides. Function of the artery: deliver nutrients, oxygen, and enzymes, hormones, whatever need to be delivered to from the heart to the periphery. Arteries, and because the artery from the heart through the entire body deliver blood that pushed by the left ventricle once every minute, or every heartbeat, sorry, once ever so often, what I guess I want to say. Therefore, there is a difference between structure of the arteries that directly associated or close to the heart with the ones that farther away. So we have three actually types, histological types that reflect function of the three different levels of artery. First, elastic artery. Elastic artery starting with aorta, large arteries. They call conducting artery. They sort of they have a lot strong pronounced elastic fibers, just like shown here. It's the same picture because it's very well shown here, that inside of this tunica media, inside the muscle, we have this pronounced layer of elastic fibers. What happened is with those blood vessels, they need to maintain their cylindrical shape, they need to be able to hold a lot of blood that comes from their left heart, and they have to push it to periphery. However, the contractility is important, but it's also important the elasticity, ability to expand the size under pressure of the blood that going from the left heart. Push it down, and therefore elastic fibers of these muscles are represented pretty well, and the name comes from elastic. It's also, so there is not so much pulsation as to contraction and relaxation of the arterial wall, but rather smooth pressure going to the smaller subdivision of arteries, which we call muscular O distribution, because farther away from the heart, when the heart force already done and over with, we need something in addition to which we achieve by smaller size muscular artery contracting and pushing, distributing blood further to the tissues. And the last subdivision is arterioles. Arterioles, the smallest one, they practically live to the capillary bed or transit to capillary bed. They have very little tunica externa. They practically fused, forming the sort of little tunnels in their tissues. They're small and mostly endothelials. Here is not folding one endothelial layer of the endothelial of tunica intima is present. They are small muscular arteries, but the muscles mostly used for like one layer. It's mostly used for extension, stretching, and bouncing back, because they mostly work of the sphincters function that we'll talk about. This is the size of the muscles that, of the different size and type of the arteries. Elastic artery, everything is there, muscles and elastic fibers. Muscle mostly represented by muscle tunica media, and arterioles don't have adventitia, have thin, very thin muscle layer and very thin endothelial layer of tunica interna. In diameters, artery could be, therefore, they different, right, in diameter. They can change diameter depending on the endocrine or nervous stimulation. And everything, contractility, changes in diameter, type of artery generate resistance to blood flow, right? So if it's well contractile, well stretched, and smooth and nice, it can generate less, and the diameter is large enough, generate less resistance, as opposed to tiny with not too very smooth surface. It will generate more with small diameter, much more resistance. What else happened, depending on the resistibility or elasticity of the blood vessel wall, we can end up forming aneurysm. Aneurysm is a bulge or expansion of the portion of arterial wall which is weak and cannot withstand the pressure of the blood constantly. It could happen for different reasons. It could happen as a result of inflammation, for example, systemic vasculitis. It could happen genetically. Aneurysm could be formed in one of the blood vessels, coronary, and aneurysm of aorta, abdominal aorta, sometimes very serious complication. The danger is, of course, rupture and bleeding. And of course, it's all very serious consequences if it's not immediately diagnosed and taken care of. It could be instantaneous death. Diagnosis is no problem as long as doctor said mind to the possibility of aneurysm. It's no problem to diagnose. The surgery was dangerous because if say brain, the surgery on the aneurysm of one of the brain blood vessels always has consequences of damaging portion of the brain. Aorta, the same thing, it profuse bleeding and death from the loss of blood. So there are very serious consequences for the situation. Rupture could be provoked by any possibility of changing pressure, such as plane, flight, altitude change, stress, anything could provoke rupture as soon as pressure at this area is changed. This is capillary. We talk about arterioles being smallest blood vessels and being delivering blood, practically not having tunica adventitia, being practically tunnels or canals inside the tissue. Here is capillary continuation of the arterioles, or the smallest transitional point from arterioles to the capillary bed. Capillary bed is the totality of entire capillary in any piece of tissue coming from one arteriole and joining in one venula. Capillary composed of basement membrane and one layer of endothelial cells, and they also consist pericytes. Pericytes are modified contractile stem cell. The function of the pericyte is first of all to accommodate, there is no muscle layer, nothing in the capillary. So the pericyte first of all accommodates the contractility and stretching motion, right? Help at least. Second, the stem cell, meaning that they're capable to differentiate in any tissue. In the process called angiogenesis, which is development of the new blood vessels, pericyte very nicely could respond to vestibular endothelial growth factor. No, I misspoke, sorry. Vesicle endothelial, vessel endothelial growth factor, and by responding to this factor, they get to develop in the new endothelial cells of the blood vessels newly generated. The blood vessels endothelial cells also capable of so-called chemotaxis, movement towards different chemicals, including vesicular endothelial growth factor and other chemokines. So the whole exchange function belongs to the capillary. However, if you think about capillary bed or capillary network in different areas of our body, such as brain, kidney, gastrointestinal tract, skin, you will understand that the function of capillaries, that all gas exchanges, nutrient exchanges between blood and the tissue that capillary support and provide the ability to exchange. Those tissues are all different. Requirements are different. For example, for brain, you have blood-brain barrier. So the accessibility for foreign substances to come to the brain should be limited. For respiratory membrane and for a kidney membrane, urinary tract membrane, the ability of capillary should be slightly expanded, but again not to let cells or proteins get out, or large molecules. Absorption in the small intestine or tubular apparatus of the kidney, again, yet another function. The parenchymal organs such as blood, liver, bone marrow, and spleen, where not only gas exchange occurs but cells exchange occurs, should be totally different, because here you have to have capillary network that allows the whole cell or let alone the protein go through. All those differences in function account for different structure of the capillary. Again, we have the same concept of structure accommodating function. So now you need to know the structural types of the capillary, and we have three of those. So we have three types of arteries, elastic, muscular, and arterioles. Now we have three types of capillaries. We have continuous capillaries, we have fenestrated capillaries, we have sinusoid capillaries. So what happening? All of them accommodate different function. In the skin muscles, where you don't want huge exchange, you need exchange of gases, you need exchange of the ions, not something that dealt with large molecule glucose, maybe. So, tight junction between the cells, between endothelial cells, rare gaps between the cells called intracellular cleft here, and nothing more. So this is basically brain, skin, muscle, where you need that tight barrier between tissue and the blood, not to let something in. Thymus is the same thing, because thymus, we will learn later, the organ where the blood cell or immune cell develops. So before they develop and get educated, you cannot take chances and bring there any foreign substances, right? Because there is no protection then. So the periocytes here, the endothelial cells here, intracellular cleft, tight junction, and everything is here in the continuous capillary. Fenestrated capillary, fenestra is a hole, so those guys have a hole in the endothelial cells called fenestration of fenestras, and this is the holes. They have also those intracellular cleft, more present than continuous capillary. They participate in filtration function, so the small intestine, endocrine gland, kidney, all this has to come with fenestrated. A choroid plexus of the central nervous system. Remember those icicles we talk about, choroid plexus that produce cerebrospinal fluid, hanging down in the roof of the ventricles. Also have fenestrated capillary, because they need to be able to give nutrients and large molecules to the brain cells. The intestinal tract, very good for absorptive function, those fenestrated capillary. And this is there, and you see those holes, fenesters that form them. Sinusoid capillary. Sinusoid capillary have large holes, large fenesters, large pronounced cleft, intracellular clefts between the cells. So where are they? They are in parenchymal organs such as liver, spleen, bone marrow, adrenal medulla. They also allowing the cells, specifically phagocytic cells, erythrocytes, when erythrocytes are going, getting cold and need to be removed. As any toxin, the molecules of toxin could be different, so any toxin can go through, and liver is a detoxicating organ, so it's all works on the sinusoid capillaries. Large lumen also account for ability to hold large molecules in cells. This is sinusoid. So they similar to fenestrated, but holes in intracellular clefts much larger and tight junction which exists but still more rare compared to fenestrated, let alone continuous. Venules, on the other hand, don't have these differences, and venules mostly are represented by a layer of endothelial tissue with gaps between adjacent endothelial cells, which made the this is a sinusoid capillary, but I was thinking about venules. And so it's similar to sinusoid, but not in such a huge cleft. There is nothing to go through. Now, capillary bed or capillary network, if you wish, whatever you wish, it's still capillary network. It's created in every portion